Hi, this is Sahir from Easy Peasy, and today we are going to discuss the topic called nucleic acids. Nucleic acid is a very important part of a living organism. So I'm taking a human being here, and when I take a closer look at that, I can see that human being is made up of cells. When I jump into the cells, I find out that, oh, there is a whole new world there. I'm not concerned about anything else. I'm just focusing here on nucleic acid. So I can see nucleic acid is present in mitochondria, in nucleus, and in rough endoplasmic reticulum. The concentration of nucleic acid is more in the nucleus, so I'm jumping into that. And I find out that it is made up of chromosomes. So chromosome consists of the nucleic acid ribbon wound around a protein named histone. I'm not concerned about histone right now because that is a protein. I'm just concerned with the nucleic acid, the ribbon structure here. So I'm going to unwind it now. So when I unwind it, I find out that nucleic acid is basically a polymer made up of a monomer named as nucleotides. And nucleotide consists of three parts here. That is a phosphate group, a pentose sugar, and a nitrogenous base. When I take a look at the other living organisms like amoeba, I can see the nucleic acid present there. When I take a look at the plants, I find out that the DNA or the nucleic acid is present in the chloroplast there. When I take a prokaryotic cell, the nucleic acid was present there in the form of a chromosomal DNA and plasmid DNA. And when I take a virus, the nucleic acid was present there as well. So nucleic acid is a really important part of living being, and it works like a brain there. Okay. Now, to understand the structure of nucleotide is really important for us. So we already know nucleotide consists of three parts, that is a phosphate group, a pentose sugar, and a nitrogenous base. Now let's discuss the phosphate group first. We can see the phosphate group consists of a phosphorus atom surrounded by 4 oxygen atoms there. Now phosphorus belongs to group 5. So it either needs to make 3 bonds to get stabilized, or it either needs to make 5 bonds to get stabilized. That's why in this structure you can see that phosphorus is making 5 bonds there. 3 with 3 oxygen and 2 with 1 oxygen there. Oxygen, on the other hand, belongs to group 6. So that's why oxygen is making 2 more bonds to get stabilized. If you remember the octet rule, or having 8 electrons in the outermost shell, then you will understand what I'm trying to say here. Fine? Okay, so now take a look here. Phosphorus is making 5 bonds. So 1, 2, 3, 4, and 5. Bingo, 5 bonds are there. Oxygen needs to make 2 more bonds. So 2 bonds are made here. This oxygen is stable, no more charge on it. These 2 oxygens have a charge, a negative charge here, because it is just making 1 bond with the phosphorus atom, right? And this oxygen atom doesn't have any charge because it is again making 2 bonds here, one with phosphorus and the other with the pentose sugar here. Okay, so that's the phosphate group we have. The bond that is connecting this phosphate group with the pentose sugar is called ester bond. Fine. If I remove this phosphate group from this nucleotide, then a pentose sugar with the nitrogenous base is called nucleoside. But once this phosphate group is attached to it, then this nucleoside will become nucleotide for us. Fine? Okay. The next thing we need to understand is pentose sugar. Pentose, the word tells us that they have 5 carbons in it. Let's count. 1, 2, 3, 4, 5. The important thing here is the 5th carbon is not present inside the ring. It is present outside the ring. So this is the really important point to understand here. Okay. Now we have 2 most famous pentose sugar in the nucleic acid, that is ribose sugar and deoxyribose sugar. The word is telling you what's the difference. Deoxyribose means that you're removing an oxygen atom from a ribose sugar, right? And where we are removing that oxygen atom, we are removing it from carbon number 2 here. Okay, that's why we have two different kind of nucleic acids, that is DNA and RNA, because of these sugar atom. So if you're using deoxyribose sugar in the nucleic acid, then that nucleic acid will be called DNA, like deoxyribose nucleic acid. And if you're using ribose sugar in the nucleic acid, then that will be called ribose nucleic acid or RNA. Sounds good? Okay. The third thing we need to understand is the nitrogenous base. So let's take a look at that one. Nitrogenous base. Why we call it a base? Because it donates electron just like a base. So that's why we use the word base with it. And why we call it nitrogenous? Because we have two different kinds of nitrogenous bases, that is purine and pyrimidines. And we can see that nitrogen atom is basically embedded in these structures. That's why it is called nitrogenous bases. Okay, now let's discuss and memorize the structure for pyrimidines. Pyrimidines consist of three different structures, that is uracil, thymine, and cytosine. Uracil is mostly present in RNAs, while thymine is mostly present in DNAs over there. Okay, now before we need to memorize the structure for pyrimidines, we need to remember this. Carbon belongs to group 4, so it will make 4 bonds. Nitrogen belongs to group 5, so it will make 3 more bonds. And oxygen belongs to group 6, so it will make 2 more bonds. Okay, I will tell you how this thing will come in handy while memorizing the structure for pyrimidines. Okay, now let's start. Okay, to remember the structure for pyrimidines, we need to find the common things present in all these three structures. So the first common thing I can see here is all these three structures are in the form of a ring and have 6 corners in it. So I'm putting that. Then I can see that nitrogen is present at position 1 and 3 in each circle. Okay, so I'm putting that here. All other sides are covered with carbon atoms, so I'm putting carbons there. The third most common thing I can find here is this carbon is attached with oxygen atom with a double bond, and this carbon is present between these two nitrogen atoms. So this is common in every structure. I'm putting it here as well. The last common thing I can see in all these three structures is these two carbon atoms are attached with double bond here. So I'm going to put a double bond here as well. Okay, so these are the similarities in all these three structures. For differences, I created a code to remember. Okay, the word pyrimidine gives me a look of pyramids, and pyramids were old and it was an ancient era. So the code I generated to remember the differences of all these three structures is this. Uniqueness of the old clause can be needed. Now the word that is highlighted here is basically telling you what to do. U stands for uracil here, and O stands for oxygen on the top. T stands for thymine here, and O stands for oxygen on the top. C stands for carbon on the side. Again, the next C is telling you that this is cytosine, and N is telling you that this is nitrogen on the top. Fine? Okay, now we put all these things together. Just by memorizing this code, you can remember the structure for pyrimidines. Bingo! Now what about the hydrogen atoms, right? Let's take the first thing that I ask you. Now we are going to use this, okay? The oxygen molecule or atom, the oxygen atom on the top, it needs to make two more bonds. Two bonds are made, right? So we don't need to attach any more thing with the oxygens here. That's done. With this carbon atom, four bonds are already made. Do we need to attach any hydrogen atom with it? No, we don't need to do that, right? Okay, now for this carbon, we have three bonds already made. How many hydrogen is needed? One. For this carbon, three bonds are already made. How many hydrogen is needed? One. For this nitrogen, two bonds are already made. How many hydrogen is needed? One. For this carbon, two bonds are made with oxygen. Two bonds are made with two nitrogen. Do we need to attach anything with this carbon? No. For this nitrogen, two bonds are made with carbon. How many hydrogen is needed? One, right? Okay, let's come here. We already know about oxygen, so I'm not going to say that thing again. We already know the common things. I'm not telling you that thing again. I'm just going to tell you this thing. So we attach a carbon group here, right? Carbon needs to make four bonds to get stabilized. Now, one bond is already made with this carbon, right? How many hydrogen is needed now? Three. For this carbon, do we need to attach a hydrogen atom here? No, because four bonds are already made here, right? Now, for cytosine, we attach a nitrogen atom on the top. It is attached with carbon, so one bond is already made. How many hydrogen is needed now? Two. So just by remembering this particular thing, you can understand how many hydrogens are needed to complete the structure. And here you go. You remember the structure for pyrimidine in, I think, 3 to 4 minutes, right? Bingo. Next. Next is the structures for purines. In purines, we have two different structures. That is adenine and guanine. Again, to memorize these structures, we need to find the similarities, right? Again, the first structure, the basic structure, has 6 corners there. So I'm taking that. Again, nitrogen is present in position 1 and 3. So I'm putting it there. Other sides are covered with carbons. So I'm putting carbons there. Okay. Now we have a side circle as well, right? Each carbon is attached to one nitrogen and nitrogen is attached to one carbon. So I'm going to do that like this, right? Okay. Now for differences, again, I created a code for that. The word purines give me a sense of purity, and purity is present in angels. So the code I generated to remember these two structures is all nice goals of nation. Again, the alphabet that is highlighted there is telling you what to do. So A stands for adenine and N is the nitrogen atom on the top. G stands for guanine, O stands for oxygen on the top and nitrogen on the side. Okay. Next thing is we need to understand how to put double bonds here before you put hydrogen atom, because hydrogen atom is the last part to complete the whole structure. But right now we didn't complete the whole structure because there are a lot of double bonds present there and we didn't put any double bonds right now, right? Okay, so let's start with that. Okay. This is a kind of benzene group for me, right? A single bond followed by a double bond. So I took this as a single bond. Now I'm putting double bond here. This will be a single bond for me. So double bond will come here. So I'm putting it here. Then this is a single bond for me. I will put double bond here, right? Okay. Now if I'm making a structure for adenine, double bond is good for me, right? But if I'm making a structure for guanine, because I put double bond O on the top, carbon is already stabilized. So this double bond will not come here. Fine? Make sense? Okay. Now with this double bond, we already have a double bond followed by a single bond. So I need to put another double bond here. So putting that followed by a single bond. Now all double bonds are done now. The remaining parts we need to attach hydrogen atom with it, right? And you know how to attach hydrogen atoms now because we did attach hydrogen atoms in purines, in pyrimidines before. Okay, so one hydrogen atom here because 3 bonds are already made, and as far as you can see the whole structure, right? Okay, next. Okay, now we need to understand how we're gonna attach the nitrogenous bases with our sugar atom. Okay, so if I'm talking about purines, I'm going to attach the adenine or guanine with the small circle there, and this is position 9. Okay, and this nitrogen is going to attach with the carbon number 1 here. Okay, so that's adenine. Again, the bond that is connecting nitrogenous base with the ribose sugar here is glycosidic bond. Fine? Okay. If it's guanine, you can see the structure. Just remove this hydrogen because nitrogen only makes 3 bonds, so this hydrogen should not be here. Fine? In case of pyrimidines, we will attach it with the nitrogen atom 1 in that structure. So that's uracil, similarly thymine, similarly cytosine. Fine? Okay, next. Phosphate is attached to carbon number 5, right? So this will be a 5' end for us. And then we will have OH group on 3rd carbon, so that will be 3' end for us. Okay? The bond that is connecting phosphate with the sugar, we already know that that's the ester bond. So if I take another nucleotide and the phosphate group of that nucleotide is going to attack the OH group present on 3' of the first nucleotide, and that's how it's going to make the nucleotide, the polynucleotide chain for us. Okay, now we have 2 ester bonds. One is made here and the other one is made here, so we will call it 3' to 5' phosphodiester bond because there are 2 ester bonds here. Bingo, right? Till now we made a single polynucleotide chain, right? So we should understand the properties of that nucleic acid. So if I'm talking about RNA, that is a single nucleotide chain. That's perfect. So we are going to discuss that here. If I'm talking about DNA mostly present in double-stranded form, so we will discuss it later. Now the sugar phosphate backbone, as we did saw in the structure, that it does contain oxygen atom with negative charge on it, and the sugar molecules have OH groups in it, so they have the hydrophilic properties. So by thinking of the structure, you should understand that the sugar phosphate backbone of both DNA and RNA will be hydrophilic. But purines and pyrimidines will be hydrophobic if they are present in normal pH. But if you're going to change the pH into acidic or alkaline environment, obviously the bases will get a charge on it, so the solubility in water will get increased. Okay, now let's talk about DNA. So DNA is present in double helix form, right? Okay, so the 2 chains are coiled around a common axis called the helical axis. The chains are paired in anti-parallel manner. So it means if one chain is from 5' to 3' end, the other chain will start from 3' to 5' end. Okay? This spatial arrangement creates a major groove and a minor groove there. You can see in the picture. And these grooves are the binding site of regulatory proteins. So the histone molecules over there, which was attached to this ribbon, is basically attached within these grooves there. Okay? Okay, now let's discuss the structures in detail. Okay? This is phosphate. This is your sugar atom, and that's your nitrogenous base. Phosphate group is attached with 5 carbon and 3 carbon by 3' to 5' phosphodiester bond. We already discussed. I already said that the 2 chains will be in anti-parallel condition, so you can see that they are anti-parallel to each other. Now the nitrogenous bases are attached to each other with hydrogen bonds. Adenine always attaches itself with thymine, and guanine always attaches itself to cytosine there. The adenine will make 2 hydrogen bonds with thymine, and guanine will make 3 hydrogen bonds with cytosine. That's fixed. It cannot be changed, right? Now the question is why it can be changed, right? Why cytosine is not making bond with thymine, or why guanine is not making bond with thymine, right? Let's discuss the structure here. Again, I'm taking 2 polypeptide chains. You can see the 5' to 3' end, and then 3' to 5' end, so they are anti-parallel. You can see that. Okay, now here you can see the cytosine is making 3 hydrogen bonds with guanine, right? So these 3 bonds are there. And thymine is making 2 hydrogen bonds with the adenine. First of all, cytosine and thymine are smaller than guanine and adenine. So these 2 structures cannot make bonds with each other because they will be far apart. Guanine cannot make bond with adenine because these 2 structures are really bulky, and inside the DNA these 2 structures cannot be compatible with each other. Fine, so they cannot make bonds with each other. Third question was why thymine is not making bond with guanine, right? Now look at the structure. In thymine we have an oxygen atom here, while in cytosine we had the nitrogen atom here. Now, hydrogen bonds can be made between hydrogen and oxygen, that's fine. But if I put another oxygen atom here, these 2 oxygen will have same charge and they will start repelling each other. So they are not compatible with each other. Fine. That's why cytosine always go with guanine and thymine always go with adenine there. Okay, next is the function of nucleic acids. We have DNA and RNA. For DNA, the major function of DNA is storage and transmission of biological information. But for RNA, we have 3 different kinds of RNA. So if you have ribosomal RNAs, the function of ribosomal RNAs is to synthesize protein. If you have messenger RNA, that is mRNA, the function of messenger RNA is to carry the genetic information from one or few genes to ribosomes for protein synthesis. And if you have transfer RNA, the function of transfer RNA is to transfer amino acids to ribosomes. Okay, so that's the basic function of nucleic acids. Now let's start the 5-second quiz to see that did you memorize the structures I asked you the way it should be, and did you get something from this video or not? Okay? So let's start. Get ready. My first question is it a ribose or deoxyribose sugar? 5, 4, 3, 2, 1. This is a ribose sugar because we do have oxygen on carbon number 2. Okay, next. Next is, what is the name of the bond present between phosphate group and pentose sugar? 5, 4, 3, 2, 1. Ester bond. If you said 3' to 5' phosphodiester bond, your answer is right, okay? And if you just said ester bond, that is right. So both answers are right. Okay, next. Next is, what is the name of the structure? 5, 4, 3, 2, 1. Okay, this is cytosine. Remember the N here? That is telling you this is the N on the top. Okay, what is this? 5, 4, 3, 2, 1. This is thymine because it does have 2 things here. One is oxygen on the top and carbon on the side. So the old clause is fitting here. Okay, next. What is the name of the structure? 5, 4, 3, 2, 1. This is adenine. Fine, because nice is on the top. Fine. Thank you very much. If you like my video, just hit the button like there and share it, and please subscribe my channel. Thank you. Bye bye.